Very interesting to see the other side of the aging field, not just the scientific part. And I have the great opportunity now to share with you what we do as vascular biologists to contribute to this research in the aging field. And our goal, as it has been said a lot yesterday, is not more to increase the number of years that a man or a woman will live on Earth, but more to decrease the angle of this curve that correlate our chronological age to the biological age, so that we will be able, as you mentioned earlier in your introduction, you will be able to feel productive more and to have a healthier aging for more years. So we have heard a lot about the effort that has been put to identify these hallmarks of aging that are pathways that manifest during aging in all cells, and if you manipulate them, you can impact the aging process. And it was wonderful yesterday to see how the biotech companies are taking over and are really producing drugs that target each of those hallmarks of aging, and we will hear about cellular senescence in a moment, or mitochondrial replacement to correct the mitochondrial dysfunction, and we have all heard about caloric restriction, CL6, etc. So in our lab, we have kind of zoomed out from these pathways that happen at the cellular level to look at the aging process at the organ level. And we all know, just by looking at ourselves, we all age, that the pathways, these hallmarks of aging, are not impacting the cells of our organs in the same way and at the same time. For example, we are losing, passing the age of 30, we are losing 1% of our lung capacity from the age of 30, but our kidneys are more resistant, and we will sense a loss of function just after age 50. And hopefully our brain is the most resistant, and we will feel his loss of function past the age of 70. So the question is, if we, so it implies that the hallmarks of aging are impacting all these organs in different ways and at different times, and targeting these pathways, the hallmarks of aging, should be according to these different clocks impacting the different organs. So, as I told you, I am a vascular biologist, but I think you don't have to be one to appreciate how this huge organ that is shared by all other organs in the body really support the function of the other organs in the body. And not only because these are beautiful tubes that bring the blood and all the blood-borne substances, oxygen, nutrients, take out the waste and bring also the hormones to the other cells, but also because the endothelial cells, which are the epithelial cells that are lining the interior of the blood vessels, are producing endocrine factors that are specific for each organ. An endothelial cells in the pancreas will secrete factors specific for the pancreas, which are different from the factors secreted in the liver or in the skin or in any other tissue. And these endocrine factors have been shown by us and by others that they are essential to maintain the homeostasis of the organ by maintaining the stem cells and also by instructing the organ regeneration after injury. When a cell is injured and dies, endothelial cells will secrete factors that will instruct the rest of the tissue to proliferate and to fill the gap. And what is aging if not losing these two capabilities, losing the regeneration and the homeostasis capabilities? So our working hypothesis was that Aging of the vasculature should impact the function of all other organs, and if we could find a way to prevent or to delay vascular aging, we might be able to obtain a kind of holistic geroprotection. So how our blood vessels age, we all have heard about the classical cardiovascular disease, which affect the large blood vessels, and remember that a man or a woman is aged as his arteries. And we all know about this arteriosclerosis, which is a disease affecting the arteries, where the wall of the arteries become stiff, and they cannot vasodilate or vasoconstrict to induce an adequate blood flow, or atherosclerosis, which block the flow in the arteries, or aneurysm, etc. All these classical cardiovascular disease is a disease of tubes, which are not able anymore to distribute the blood to the other organs, inducing ischemic injury in the end organs. But we also know that these classical cardiovascular disease, we have plenty of ways to correct them. Behavioral changes, nutrition, exercise, sleep, avoid the smoking and drinking. But we have also a battery of drugs that were shown to alleviate or to delay or even suppress the onset of these diseases. And for example, rapamycin, metformin, velbesterol, NAD precursors, all these drugs that impact in a positive way the large blood vessels are also anti-aging drugs, right? But what we were interested more in the lab is about the capillaries, not the large blood vessels, but the tiny blood vessels that are the ones that are in contact with our cells in the body. And we don't know much about how these blood vessels, these capillaries, are aging, but the most prominent phenotype is their disappearance. When we age and we observe tissue sections of aged mice or aged human, we could see a decrease in the microvascular density. And this shouldn't happen, because in each of our cells we have a mechanism that earned the Nobel Prize to this scientist in 2019 for discovering this pathway. When any cell in the body will sense a lack of oxygen or nutrients, it will switch on the HEAF pathway that will induce the secretion of factors which will instruct the neighboring blood vessels, capillaries, to grow and to bring back the perfusion to the ischemic tissue. And one of these factors is very interesting, the vascular endothelial growth factor, because it is involved in all aspects of the angiogenic process. And also it is involved in all the specific features of endothelial cells in different organs, including the secretion of these specific angiographic factors. And most importantly, it is indispensable to maintain an adequate capillary density in our tissues. And for example, if we block VEGF signaling in a 4-month-old heart of a mouse, we will see that a decrease in the microvascular density that will be restored only when we will restore the VEGF signaling. So there is no backup to VEGF. So this was a great candidate for us to look at to see if it has an impact on the aging process affecting the blood vessels. And what we saw that in aging organs, both in humans and mice, we have a decrease in the VEGF signaling pathway that we measured by measuring the phosphorylation of its principal receptor. And it is not because a reduction of VEGF, we can measure VEGF in the circulation or in the organ of the mice. And by the way, we got the same trend in the human, in the analysis done in the blood of human. This is the analysis done by Benoît Lehallier in the lab of Tony Viscore. And if you could see that the last cohort that behaves exactly like our aged mice is the centenarians. It seems that centenarians have more VEGF in their blood. So it's not a problem of the ligand, you have as much VEGF that you want, but we could detect the accumulation of an inhibitor of VEGF, which is the extracellular domain of one of his receptors, which is produced by alternative splicing. And it sequesters VEGF and do not allow signaling of VEGF. So the question that we asked was, can we maintain, is there a way to maintain VEGF signaling in aging mice? And we could do that by targeting, of course, the inhibitors, but I just presented you one inhibitor. It seemed that we saw that we have lots of inhibitors of VEGF that are accumulating with time in aging. So we decided to saturate the inhibitors by increasing the level of the ligand, the level of VEGF. Oh, we do that genetically by manipulating our mice, and we could obtain twice the amount of VEGF, which is generally produced by normal mice during aging, when they age, and we could elevate the level of VEGF twice the amount of naturally produced. And this dosage is very important because in our system, if we go too high on VEGF, we have deleterious effects such as edema and an impact of the hematopoietic system, but this is also an interesting story, but for another time. And pay attention that we begin this treatment at about 10 months of age of the mice, so they are not very aged, they are not ill, so all what I will present to you is about preventing the age-associated disease and not reversing them, as Chaim Cohen presented us with the CIT6 story. So when we did that, we could maintain a useful amount of VEGF signaling in the tissues of our mice, and the consequence was to maintain an adequate microvascular density in the liver, in the muscle, and all in other tissue, brown adipose tissue, white adipose tissue, bone marrow, all the tissue that we have looked at in these mice. And what was the consequence, the first consequence of this maintenance of perfusion? Well, it's oxygenation, right? So we measured the oxygenation of the tissues in these mice, and we could see that it is completely maintained as the level that we could see in young mice. So we follow a cohort of 100 mice, female and males, treated with VEGF and control mice, which are little mates, and we could see that not only we were able to extend the lifespan, that was not our goal, but we also compress the morbidity period. This time when the mice begin to die because of these age-associated diseases and frailties. So we went after all the phenotype that we could measure in these mice in our lab or by way of collaboration. And because this has been published 1 year ago, 1.5 years ago, I will just summarize the results. We looked at the distribution of the adipose tissue, which is something that we could see on ourselves when we age. Aging is about fat, I'm telling you. So we could see that the abdominal fat that is generally accumulating in aging mice is alleviated in VEGF-treated mice. And the contrary which happened in the skin, where we lose our subcutaneous fat, is alleviated also in VEGF-treated mice. We also looked at the brown adipose tissue, the white adipose tissue, and I will just run because I'm going out of time. We looked at the metabolism, we looked at the liver, accumulation of fat and injury of the liver, we look at the muscle, we look at the bone. We were amazed to see how much the volume of the bone is preserved in these VEGF-treated mice. And maybe because of good muscles and good bones, we could see also a better kyphosis index that we all see in our grandparents. And I will just take a few minutes to expand a little more about phenotype that that we have seen in our mice is about the tumor burden. Because when we say VEGF in an audience, immediately you connect it to cancer, right? Because cancer cells are producing tons of VEGF when they grow to recruit new blood vessels, so that can proliferate and increase their volume. And when we followed the mice by MRI, we could see that if the 24-month-old mice had a lot of spontaneous tumors, as you can see here, VEGF mice were protected. And when we followed the mice at time of sacrifice and we observed how many tumor lesions they had, even at a very old age, our VEGF-treated mice were tumor-free. And now we are pursuing this aspect of this research, trying to understand why we have less tumor when we have better blood vessels, better capillaries. And one of the directions is to show that we have less inflammation. This inflammation, which is chronically present in aging mice and in aging humans, in VEGF we can see a decrease in this inflammation. And by the way, it's one of the hallmarks of aging that we correct with VEGF treatment. We also looked at the senescence. We didn't see much of senescent cells in many organs, but in the liver, it was astonishing to see that the endothelial cells are the first cells to become senescent, and we could correct them with VEGF treatment, which implies that VEGF can protect the endothelial cells from entering the senescent process, or maybe impact on the immune system that can remove in a better way these senescent cells. And just to cut a story long, done by Priya Gupta, a postdoc in the lab who is sitting in the audience, we also found that the immune system of these mice are improved. The thymus is not undergoing the atrophy that is happening in all the aging mice and the human. We have more naive T cells, and we are very excited to see that the T cells that are in the mice treated with VEGF express much less of the PD-1 that we have heard about at the immune checkpoint, which can explain why we have less tumors and why we have also less senescent cells, since these cells have been shown to express this PD-1 ligand. So I stop here, and sorry for taking a little bit of more time, just to thank the people in the lab that have participated to this study. Of course, Professor Eli Keshet, that I'm working with for many years now, and the postdoc and the PhD student that are looking at different aspects of the aging process. And we are recruiting at the level of postdoc, so if you are interested, please contact me. And this, I was always wondering if the fact that VEGF signaling pathway has entered the conscious of the aging field. So I asked yesterday night the chat GPT what he thinks about VEGF and aging, and that's what goes out of it. So I think that it is in the conscious of people. If you have questions.
Very impressive results in mice, and you did mention that also there is an effect in humans. Can you say a bit more about what the next stage is for human trials, possibly?
So, for a translational point of view, we just got a grant with a company, Merck company, who is now supporting us to develop alternative ways, which are not genetic, to increase the VEGF signaling. The first thing that we are going to do, and we have begun already, is to produce a AAV, adeno-associated virus, which is FDA approved to transiently express VEGF. What we have found also is that you don't need a constant expression of VEGF to get this effect. We have done preliminary results because it's enough to get higher VEGF for 2 weeks to build stable new capillaries. So we have really a good hope to be able to translate these amazing results. You're right, because it was really amazing to see how these mice are doing better in everything that we looked at to human trials. Yeah. Yes.
Thank you very much for the presentation. It was wonderful and it's quite inspiring. But I'm a clinician and I have one practical question about what about the retinal vessels and choroidal vessels? Thank you.
So you are right. You have all these diabetic retinopathy, which part of them are due to over secretion, creation of blood vessels. We looked at the retina of our VEGF producing mice and we couldn't see any effect. Maybe because the VEGF that we produce is from the liver, and that's what we intend to do also for human translation. And maybe because of the blood-brain barrier, this doesn't impact the blood vessels of the retina, behind the retina. But this is a point that we looked at. What is important to say that you know that people are treating patients with AMD, wet AMD with Avastin, right? And it is now shown, because this is used for many years now, that it has an impact of the blood vessels, of the capillaries perfusing the retina. And it seems that the prolonged use of Avastin destroys the plexus, which is something that we are pursuing with an ophthalmologist in Hadassah to see if prolonged use of Avastin can impact the blood vessels, the normal blood vessels of the retina.
Hi.
Okay, next question, please.
Oh, may I ask, so I was just wondering, you're talking about the antibodies, that this real problem is there's antibodies being produced against VEGF, and now you're giving more VEGF. And I'm just wondering, is this like a symptom of autoimmunity disease? And would that be actually the way to tackle this? But go ahead.
Yeah, so it's not antibodies that are produced, but a soluble form of the receptor of the VEGF, which is produced, but what we call alternative splicing. Generally, you have the RNA that encodes for the whole receptors, but with aging, there is a mess in the alternative splicing machinery, and the extracellular domain is produced and is secreted in the blood. And this, which is done in normal situation, is exacerbated in the aging mice and human also. And this is sequestering the VEGF and inhibits its signaling. So a way, you're right, there is a way to inhibit this inhibitor. The problem is that we saw that in the blood of human and mice, we have other inhibitors. So if we want to target the inhibitors, we will have to have a battery of antibodies or small molecules that will inhibit each of these inhibitors, and this is less feasible.